Niko, ¿se va a acabar? ¿Se va a grabar? Just to let you know that the broadcast it will be broadcast to the internet. Bienvenido todo el mundo. Welcome everybody. This is the defense by Niko Komin, directed by Raúl Toral. And okay, the defense will proceed by your presentation. There is no official legal limit for your presentation, but will be considered polite to be under the 1 hour limit. And then the committee will ask you whatever the committee thinks it should be asked. So, go ahead.
Okay, thank you very much. Welcome everybody. Thank you for being here. Thank you for the committee. And so I will present my PhD thesis with the title Phase Transitions Induced by Diversity and Examples in Biological Systems. It was done here at the IFISC, as you said, under supervision of Raúl Toral. And well, financial support came first from the Biosem network of excellence, and secondly from the government of the Balearic Islands. So to start this talk, I want to give the motivation of this work. The idea was that in biological systems, the assumption of identical units does not hold, as for example, with considered in atoms or electrons. So in biology you won't find any cell which is the same as another cell. And to put the main result in front of, more or less the main result in front of the whole talk, is that macroscopic order can be induced by diversity, or diversity as it is called as well, microscopic disorder or quench noise. And the outline the talk will be divided in 4 topics, more or less. The first is a model of drug absorption. Then I will talk about circadian rhythms, active rotators, and in the end about phase transitions induced by diversity in a more general manner. So the whole investigation, as it started within the Biosem network, which was a conglomeration of European scientists from different fields. I want to present the purpose of that network. It was meant to develop in silico simulation models. In silico means numerical in computer models of cellular physiological and pharmacological processes to understand diseases and the therapies, understand them better and devise better therapies from that. So we started by investigating a model of drug absorption. So imagine you want to take a medicine for some illness, and there's a variety of ways to take this. One of the more comfortable ways would be to take a pill or a potion, and these substances usually are absorbed into the body in the small intestine. And if one looks at the small intestine, which we see here, a cross section, then it is interesting to notice that this kind of tube with twines in the wall, the wall is made up of one single level of epithelial cells. So the drug, which has to get into the bloodstream, here's the capillary. So here, to get from here into the bloodstream, has to pass through this one single layer of cells. And as we know, all of the cells are, the walls of the cells are composed of these molecules that have a hydrophil head and a lipophil tail, and then submerged in water they can form different stable structures. here we see some of them. When they close, when this layer closes up on itself, we have a liposome or the cell on itself, and the substances have to pass as a first barrier 2 times this membrane. These membranes, they have the ability to embed proteins, larger molecules, depending on their consistent or on their properties. They can be embedded here and can have a variety of different functions, very different functions. One, for example, of the so-called tight junctions. They are proteins embedded in those part of the walls in the epithelial cells, and they hold these cells in place for once, and on the other side they prevent larger molecules to pass along the gap between those 2 cells. And this is one form of proteins. There's other proteins which will be more on the topic of what we see in a minute. This so-called transporter proteins. They the original function is to get toxics out of the body or out of the cell. In most cases, these medicine or the substance is considered, the medicine is considered as a toxic from the body, so it is actually expelled, and this then limits considerably the amount of drug which gets into the body of the patient. And it is responsible for the resistance of cancer cells to a variety of drugs. So it is of large interest to know how these transporters work. Anyway, if you develop a new drug and want to see one thing is if it works good or not. The other thing is to know how fast does it get into the body, how much does it get into the body. So scientists do experiments. They take these cells and seed them on a layer in a small pot, and then they have experimental conditions where you have an upper volume, a lower volume, and put the substance in on either side or on one side and measure the concentration on the receiving side. You do that on both sides for different initial concentrations, and then the slope, this is the first 2 hours here, the slope of the first 2 hours is what is called the apparent permeability in one direction and in the other direction, and the ratio of those 2 numbers gives in the end tells us how strong this substance I'm investigating is affected by this nonlinear transporter I was talking about, this active transporter protein. To get from these data to the parameters of the absorption parameters of the substance, a model is devised. This model takes into account a few assumptions, and so if we want to write down the equations for the mass of molecules in the volumes A, C, and B, then we assume first that in the upper and in the lower volume, as this in the experiments, this pot is stirred quite a lot. We consider that the concentration is homogeneous in the whole volume. And then there's another assumption which is much stronger than the first. It supposes that all of these cells have exactly the same parameter, the same size, and so on. And then one can consider the sum of these volumes as 1. So we end up with a 3-compartment model, which is actually not only used in absorption studies, it can be used as well in the movement of substances from one organ to another via the bloodstream and so on, but we will keep that absorption study. And these equations are made up of one part which is linear, which is only the passive diffusion across the membrane, and then there's this other part, the nonlinear part, which represents this transporter protein. One considers that the mass is conserved. That means that this set of 3 equations can be reduced to 2, and or the degrees of freedom are 2, and then that means that we, in this case, we were able to take this system, do a linear transformation of the concentration into a picture from classical mechanics. That means that we have an equation for a ball and a potential, and this ball is affected by a friction force. Now, both the force of the potential and the friction force have parts have linear parts, and then they have the nonlinear parts from the pump. So we need to solve these equations. We need to do some approximations, and then so we did 2 approximations on that. One is the potential to use a harmonic potential instead of the full potential. The other one is to approximate the friction by a constant, and we take the constant of the friction at the equilibrium concentration. With this, we obtain a linear equation. This linear equation of second order, we know that it has two, that the solution is a sum of two exponentials. And if we then undo the transformation and calculate the evolution equations for the other two volumes, we end up with three equations, and all of them are the sum of three exponentials. So having three exponentials means that we identify there three different timescales of this process. And these different timescales, we can express them in terms of the parameters of the system. If we have them, we can directly derive them. And here we see in this expression of the timescales that there is the possibility, if this part is below the square root is negative, then it allows for the possibility of damped oscillations in this absorption profile in theory. The other quantities, what we were saying, the apparent permeabilities can be derived as well from the parameters, and here in this case we omitted the T2 because it is usually much smaller than the other ones, and the saturation concentration has to be expressed or found in form of the minimum of the potential. This can be done in the case of the Michaelis-Menten flux, can be given a closed solution for this. This E flux was chosen in this experiment because one considers that the transporter protein takes the molecule on one side, it has to bind, and then it is shoved on the other side and released into this space. So this is similar to enzyme kinetics, and that's why this is a way of modeling this pump. If we do so, we find this solution without the equilibrium solution, without using the approximation, and then can derive the time constants and so on. And we see if we compare our approximate analytical solution with the numerical integration of the full system, and we see that the difference is, there's hardly any difference to be seen between the dotted and the line, the continuous line. And it is in fact so on all time scales. This is for 100 hours. This is the first hour of the same graph. So we see that all time scales, the approximation is quite precise to the full solution. As I said, we find this apparent permeability from the parameters, and then this the ratios, the efflux ratio is what is the pharmacologist interested in, and then as we have these close dependencies of the parameters here for the nonlinearity, here the linear parameter of the reflux ratio, so we can draw these curves, we can write them down as a formula for different initial conditions, and then we learn from that, we can learn a few things. One is that you can calculate the error propagation, that if you have an error in one of these parameters, you know how this would be propagated into the measurement. Another thing is, one could see that, for example, here, for high concentrations, measuring this efflux ratio, this parameter wouldn't be able to be determined so precise as it is the case for lower concentration. On the other hand, we see in the last picture in the dependence of the initial concentration, the blue dots are the measurement, the data from the experiment, from Valencia, the group, I didn't say that. And we see that for low initial concentrations, it doesn't really, the model doesn't really describe what is happening in reality. But this is not consequence of our approximation, it is consequence of the model itself. So to conclude that part, the first part, we have given an approximate solution, or an approximation which gives us an analytic solution of nonlinear three-compartment models, which are widely used in pharmacology. We can identify timescales, 3 in this case. One of the timescales allows for damped oscillations. and in the case of a Michaelis-Menten flux, we can give a closed expression for the exact equilibrium concentration. And now, having done this investigation within the environment of the biosym network, we learned that there's many drugs, apparently especially cancer drugs, that are the efficiency of the drug depends on the moment in the day when the drug is taken. So in fact, this leads to what is called the chronotherapy, which supposes that the moment when the drug is applied to the body is relevant to the whole therapy, and it is investigated what would be the perfect moment. So thinking of this, this is probably a consequence of the different hormone levels. For example, the hormone levels in the body, which can change during the day, or the body temperature. But interesting is that most of these body functions, they are periodic from day to day, and this period from day to day is called the circadian rhythm. And here we see now this circadian rhythm is what controls the sleep and wake cycle, the moment we feel hungry and all these things. And it has been shown that the master, well, there's a few clocks giving these rhythms in the body, but the master clock is in the so-called suprachiasmatic nucleus, which is a part in the brain of roughly 20,000 neurons. These neurons are biochemical oscillators connected among each other, and they are in contact with the state of the body, and have a direct connection with the retina from the eye. So they have a direct information about the light outside, and this light is what entrains the phase of our days, which means that if I travel across time zones and I stay in I stay say behind with my personal rhythm, then the sleep wake and all that will be disturbed, and then the light and day change will actually entrain the phase of my personal day again to what is the day in reality. Before I come to this, I want to have a short overview of biochemical oscillations. Just to start a bit, this so-called central dogma of my molecular biology is that we consider all things. The gene on the DNA is transcribed at some rate into the messenger RNA, which can decay, and this then is transcribed into the protein, which can decay as well. And this network, say, of reaction network can be more complicated. The gene can downregulate its own expression, or it could upregulate it. It could regulate the expression of other genes, so the network can be quite complicated. One network I want to tell a bit more is the so-called Goodwin oscillator. So you have the protein here named with Y, which has to be activated to be actually functional, and then it in turn it inhibits the expression of its own messenger RNA, and this leads, when you integrate this, when you look at this with the right rates, it leads to oscillations. These oscillations are so-called limit cycle oscillations. That means that if at some point, at some time, I add some molecules of the substance here, then the perturbation will actually be in the amplitude, and this amplitude will go back towards the original oscillations, so that the only perturbation is in the phase. We see that here in the phase portrait, the closed orbit. Here we have the perturbation which spirals back into the closed orbit. Coming back to this circadian rhythms, we know that the biochemical oscillators in this nucleus are based on some derivation of that Goodwin oscillator. They now have been found more proteins taking part in this process, but here we have one possibility of devising this oscillator. So we see that it is a bit more complicated. We have more nonlinearities than the original one, but there's more differences which are more important. There's one. This is here we see that there's a neurotransmitter which diffuses out of the cell. Consider this to be within one cell, and then it just diffuses out of the cell and distributes among all these cells, comes in again, and then couples to the expression of X. We have another difference here. We have this periodic input, which resembles the light signal. And then we have another difference, which is the most important thing in what we're going to investigate. From experimental observation, one knows that these cells, if they are separated, if they are not in contact with each other, they oscillate at their own period, and this period is distributed according to, well, some distribution. It is not fixed at 24 hours. It is actually so that the natural period of this oscillator is somewhere between 20 and 28 hours. And exactly this diversity, the width of that distribution will be of interest for us. We want to see how this diversity acts on the response of the population of oscillators to the external signal. And to this end, we use this variable tau I, which rescales the period of one of the oscillators, and we will take this from the given distribution. Now, to start the analysis, we think first of taking away the sine part here and considering only a constant light source. Then we can integrate a population of these neurons with some given distribution for different diversity, and we see for rising diversity, for very low diversity, we see that we have quite regular in-phase oscillations. For higher values of the diversity, we see that the oscillations have random phase shifts between the individual oscillators, and in the last case, where it is even higher, we see that the oscillations are damped and die out into the fixed point of the system. If we now, instead of taking the constant light, we go and put in the periodic light, as one would expect, then we see quite a different picture. We have in the first case with the same diversity, we see that there's oscillations with a beating on top of the amplitude. In the second case, we have a beating and the random phase shifts we have seen before. And in the last case, we see that we have steady in-phase oscillations, and these oscillations are in phase, as we will see in a minute, are in phase with the external signal of the light forcing, and in phase and in period with them. So to characterize these different behaviors, we use two different measures. One row is measuring the internal synchrony between the neurons, how they are synchronized among themselves. It is between 1 when it is perfect synchronized and 0 when they are not synchronized. And we use the second measure, the response, or the parameter R, which is taking, in this case, x, variable x, the time series of x in this case we take x. We do the Fourier transform and measure the power at the output spectrum right at the driving frequency, which would be our 24 hours, which we put in the system. This is the two measures we will take. Then we calculate another value where we take again, we get rid of the sine function. We have a constant function. Take this set of like a population of these oscillators with different time scales, linearize them around the fixed point, and calculate the Jacobian. This, as we know, depends on the realization of these noise variables. Will be a random matrix. So from this random matrix, we calculate the eigenvalues, the real part, the maximum, and can then calculate something like the average stability of the population. And doing so, we see here for a given value of the coupling and the light, the amount of light, we see first the internal synchrony. When this diversity is 0, the internal synchrony is 1, as they are doing all the same, but then it goes down up to one point where it goes up again and passes through a maximum to then go down for higher values of the diversity. In case of the response, we see that for low diversity, the response is low, but then it goes up and passes through a maximum where the response is best, and this coincides with the more or less with the region where this row has a maximum as well. One thing to have a look at now, what can we look at is the mean of the individual periods. We see that they are first much far away from the 24 hours, and here they break down and are quite constant at 24 hours. So we see that we have some optimal response yet. If we now look at the average eigenvalues, what I was saying before, then we see that it actually passes from, at low diversity levels, we have a positive eigenvalue. That means that the fixed point is unstable, we have sustained oscillations, and then for higher values this passes to be negative. The fixed point stabilizes itself and the oscillations die out. So we see that at this point is more or less where the response goes up and the synchrony as well. We see at the here we see this parameter R, the response in a logarithmic scale for a whole range of light, amount of light and diversity. We see that in the lower corner here, this is all black, we have low response, and then it goes up to higher values in the logarithmic scale. And we see here a black line, and this black line, which encloses the region of low response, this black line is exactly the line where the eigenvalues pass from positive to negative, where the oscillations are damped and die out. So this is all the region where the oscillations are damped. So this is not the case. This is not only the case for a small range of parameters. We actually see that we can do this for lots of values of different coupling, and in all cases we see the same. We see the same thing. We see the line where the stability changes, and this is the line where the below the response is low and above the response is high. So we conclude from that the diversity we introduced into the system leads to the oscillator death, and that means that the damped oscillators are better entrained to the external signal, and that better than anywhere else. And this means that an optimal amount of diversity actually improves the response of the system to the external signal. And as we have seen, this biochemical oscillators is of dimension 4. It is highly nonlinear, and it has a lot of parameters. It might be interesting to look at a simple oscillatory system and how the diversity acts on these systems. So we took the model of the active rotators, which, to explain shortly what it is, we have this phase variable phi, and the derivative is given by the right hand side of this equation. So if omega, the natural frequency, is larger than 1, then the right hand side will be always positive and the phase will be rotating on and on. Whereas if omega is between -1 and 1, then the right hand side can be positive or negative and 0, which means in fact that we have on this unit circle we have a stable fixed point and an unstable fixed point. So the phase rests in the stable fixed point. If it gets perturbed, it comes back to the rest state, and if it is perturbed over the unstable fixed point, it does the excursion along the whole unit circle, and this is a typical behavior of an excitable system. And this is why this model is a very simple model. It can be used as a simple model for neuron dynamics. Now, coupling a population of these active rotator models, we have here this sign coupling, or the sign of phase difference, and we have this frequency distribution according to some distribution, the natural frequencies. So this model, if it wants to be solved analytically and completely, it is quite a difficult task. It is involved calculations. We used an approximate method which can be used for a variety of systems. It is based on these works from the early 2000s, and we extended it and rigorously to more detail. And it starts from the microscopic dynamics, which have these functions, and these functions can be then developed in a Taylor series around the mean value of the dynamic variables and the mean value of the perturbation, the time fixed, the random, the disorder. And averaging this Taylor series, we find the equation for the mean value, of the dynamical variables, and this mean value depends on two more dynamical variables, one the fluctuations and the correlation. So we can, with the help of these expressions, we can divide these the dynamics, and we get a closed system for macroscopic dynamics. And this system can give us as well the equation for the macroscopic fixed point. Now, doing so on this active rotator model, we find these equations, macroscopic equation for the global phase, and we can calculate the fixed points. Or we can see if there is any fixed points, and then we see, in this case, this is the equation for the fixed points. We see that if there is a global fixed point, the global phase will rest in that fixed point, but if there is no global or fixed point in this expression, then the global phase will be rotating, and this means that actually all or a large amount of the individual oscillators are rotating coherently all together. So to calculate the existence of fixed points, we see this equation. The right-hand side is periodic and 2 pi, and the maximum. So if we draw in a graphical analysis, we have here the mean value of omega, and this curve is the maximum of the right-hand side. So for low and for high diversity, we will actually find a fixed point. So the global phase will be constant, whereas in between the global phase will be rotating, and we find coherent behavior. We find an ordered state. Here in this graph, we see the phase diagram. The white triangle is the ordered state of coherent behavior. We can calculate from these equations a large coupling limit for the two critical diversities, and we see in comparison with the numerical simulations that the second transition is highly overestimated. But this result in theory is valid for general distributions as long as a mean and a standard deviation exists. In the numerical simulations, we have to decide. We took first a Gaussian distribution, and what we find is indeed that the phase velocity is 0 for high and low diversity values, and in between it is non-zero. So we find this coherent behavior. If we in this numerics, we calculate the fluctuations of the order parameter according to this formula, and we find that they peak around the transition from this order to order. They peak, and this peak rises with system size, which is a typical sign of critical phase transitions. So what we can do now, or what we did, was doing a finite size scaling analysis, fitting these exponents, these finite size exponents. When they are fitted, then one can collapse the curves of the order parameter for various system size all into one curve, and the exponents we find here, the finite size exponents in the thermodynamic limit, lead to exponents. Here for the order parameter we find 1/2. For the fluctuations we find the exponent 2, which is actually not what would be expected for a system with this mean field coupling. We see that in a minute. We see more of that. We did then a simulation with exponentially distributed frequencies. That means that the mean and the standard deviation are varied simultaneously. And in this case, again, we have disordered and disordered phase. The entrance into the order phase happens through a phase transition, a critical phase transition with the exponents that is the same as we just had before. The difference in this case is that the re-entrant transition into this order is now of first order. We see that in the histogram of the order parameter. At the first transition here, it is concentrated. First, it is concentrated around one value, and then it broadens the distribution and grows to higher values. Whereas in the second transition, it is concentrated around either of the two solutions. And the integration of the macroscopic equations from the order parameter expansion actually leads to the same result, that we find a continuous transition in the first case in the entrance, and here we find the transition of first order. Concluding this third part, we show this order parameter expansion, which leads to approximate macroscopic dynamical equations, and these then can be found from those. The phase diagram can be found. We derived explicit formula for the high coupling limit. So this order parameter expansion is a fast way to obtain, an easy way to obtain certain information. The critical exponents we found, they are not what would be expected from the mean field theory. And in this case of exponential distribution, we found that the transition is of first order. This can be the second transition is of first order, and this is predicted by the order parameter expansion, the difference of this. And now, as we have seen here, phase transitions with critical exponents that differ from what would be expected and so on. We decided to use prototype models for phase transitions. We used to analyze those phase transitions, how they are induced by diversity. So in these models, we have taken these three models. They are known to, in the case of noise, of normal noise, they are known to have the destruction of an ordered phase or the generation of an ordered phase, and in this last case, the generation of an ordered phase and then the destruction of it. So to solve these models, now with the diversity to solve these, we rely on the Weiss mean field theory. So we part from the microscopic fixed point. We see, if we look at those all of these models, it is possible to write them in the form of the derivative of the potential of a Lyapunov potential, and this potential in turn can be written as a sum of local potentials, which in turn only depend on the variable xy, xi. So in the end, this can be written as the derivative of a local potential. That means that if we find the absolute minimum of the local potential, as this, the sum of it is the global potential, we find the absolute minimum of the Lyapunov potential of the whole system. And this, so if we find the solution of the local potential, then we know that averaging over the population, we would obtain the mean of the variables, of the dynamical variables, and as the solution of them depend on that mean on itself, we have this self-consistency equation, which in the thermodynamic limit, this instead of a sum has this integral over the distribution and the solution, and then having the m on both sides, this equation can be solved numerically in an iterative way to a high degree of accuracy. Doing so now, before we do so, we have to find the solution of the local potential. We see here in these three cases that for a fixed value of m, the eta value in the first case for c larger than a, in the first case eta varies the solution in a continuous way, whereas in the second, the solution is, although continuously varying, there is at some point a pair of a stable and an unstable solution arising. So for larger values of eta than that, we would have three solutions, two stable to one unstable. In the last case, this is different. We have for very low and for very high eta, we have continuous solutions, but in between we have this range of three solutions, the unstable solution in the middle. But with these formulas, we can now solve these models. Here, the black lines are representing the order parameter expansion, but the gray line is the solution from that mean field theory, and the dotted lines are the results from numerical simulations. We see in the first part that the ordered state is destroyed by this diversity. The finite size effects we see here for rising system size, So there's typical finite size effects, and we will find here this is a critical phase transition, the destruction of that ordered state. In the second case, we have the generation of an ordered state as well through a phase transition. But this order is destroyed not through a phase transition. It is rather continuously going down towards zero, and we don't find another phase transition. In the last case, again, we have the generation of an ordered state and the destruction through a phase transition. So one can say that for all these three, for the three models, we find qualitatively the same behavior as is known in the case of normal noise. Again, we do an analysis of the phase transitions, the finite size analysis. We for the three models, and the last as well for both transitions. I only show here for one of the transitions, and we find that the first and the last again show an exponent which is not what would be expected for mean field theory. In the second model, it does bring up these exponents, but not in the first and in the second and the last. So to analyze this a bit into detail, we took this model, the first, and had a closer look. To this end, first, this is just technical detail. We rescaled to get rid of the C, and then instead of C larger than A, now we say A smaller than 1. The other case I will talk about in a second. So in this case, one can, from considerations of the symmetry of this solution, one can find this exponent for the ordered parameter, which is 1/2, and then one can as well take or calculate the susceptibility, which is the derivative of the ordered parameter with respect to the perturbing field. And in this case, we find that an exponent D, we would find an exponent of 1 in this susceptibility. Now, the so-called fluctuation dissipation relation relates the susceptibility of this with the fluctuations of the order parameter, and we found in the numerical simulations, we found this to have an exponent of 2. So there is a discrepancy between the two values, and now one can say that usually this fluctuation dissipation relation follows from the fact that the stationary probability distribution can be expressed with, say, with the help of a Hamiltonian, which is the sum of the unperturbed Hamiltonian and the part with the field, which only enters linearly into the Hamiltonian. Now, in expressing the stationary probability distribution for this problem, we find that it is not possible to write it down with an exponential, with an exponent separating this in these two cases. So in fact, it is not. One would not have to expect that these exponents are the same. That this relation is actually valid, as we are not able to express the probability distribution in this way. Now, this is only what we say for this case. In the other case, where we have A larger than 1, we find again that the local potential has this picture of having an intermediate range of having 3 solutions. And in this case now, as we have the possibility of 3 solutions locally, that means that that it is not clear in general where the system would end up. So this is we find metastable states that exist in this case even for the thermodynamic limit. So it is never clear where the system in the end ends up, because there's different minima in the potential. Another thing that we find in this case is that there's additional transitions, or there's an additional transition from disorder to order, something which we haven't seen before. You see here that the curve actually starts in a disordered state, goes into order, and then goes back into the disordered state, but before the theoretical result. And then in this phase diagram, we see that the range of an ordered state, of the ordered phase, is actually smaller than what is taken from the theoretical result. As we will see, this is a consequence of of the metastability and of the dependence on the initial conditions, since this result, the phase diagram, and this is obtained for the initial conditions to be symmetrically from negative and positive. Whereas if we only take positive values for initial conditions, this picture changes. First of all, we don't see this the beginning, the disordered state, and second, we see that this destruction of the ordered state from a certain value of A on, for higher A, this destruction is not through a phase transition. It is actually continuously going down to zero. So that means that here the region of the ordered phase is much larger than it is in this case. And the third thing noticing from this, because this initial dependence on initial conditions, we have here the positive range where we start the initial conditions. We have this delta here, and then we find that the first curve is for a small delta, and then rising the delta, this approaches this curve. So we find, even taking the positive from the positive range, we find a dependence on the initial conditions. And now a detail which doesn't show up in the thesis, as we didn't investigate it very thoroughly, but it is clear that in this model of the multiplicative diversity in the Ginzburg-Landau, this is what I showed before. This is the picture, and here as well we have taken symmetric initial conditions. Now, if this is taken to be only from the positive half plane, then we see again that although the phase is the same, it is not destroyed, the ordered phase, but it is different, the occurrence, than it is here. So we have as well dependence on the initial conditions due to the local bistability or metastability. And this, now to conclude this, the fourth part of that work, we have determined this the global solution from local fixed points through this self-consistency equation, which numerically can be resolved to high accuracy. And we found that for these models we have investigated that the qualitative effects are the same for diversity as was expected, or as is seen with noise. We found that this additive Ginzburg-Landau or 5-4 model and the canonical model for noise-induced phase transitions, they show exponents which are not the ones in mean field theory. As we have seen, at least in one case, is because the stationary probability distribution has to be expressed, or is expressed in a way which is not the usual one. And then we found for this additive model that the metastable regime shows metastability in the thermodynamic limit. It showed additional phase transitions and very strong dependence on the initial conditions. To summarize the whole work a bit, it started with this highly precise approximation of nonlinear three-compartment models, which gives access to the parameter dependence of experimental quantities, and we applied this method to the selected nonlinearity and the selected experiment. And then the pacemaker cells of the circadian rhythms, we've seen that they have diverse natural periods or frequencies, and we could show that an optimal amount of diversity improves the response to an external signal because it drives the oscillator into oscillator death. And there it is better entrained. Then we did the analysis of the active rotators with diverse frequencies, which one knows that can synchronize globally. But what we did was, apart from the order parameter expansion and this analysis, we found these phase transitions on both sides and analyzed the the critical exponents and found that this transition can be can be critical and can be of first order as well. And in the last, then I want to say that we found that these classical models, say for the phase transitions due to noise, they show the same result or the same behavior due to diversity. And another result, say, is that the 5-4 model with analytic diversity, this canonical model for noise-induced phase transition, and the active rotators seem to belong to the same universality class. And with this, I finish my talk, and I thank you for your attention.
Okay, so now the committee will ask you questions committee thinks are pertinent. So let's begin with Professor Parrotti.
Okay. Well, let me first express my congratulations to you because it's a very nice work. Also to extend the congratulations to Raúl, the advisors, and the rest of the group. It's a very rigorous work with a lot of ideas, and I like also very much the biological applications. And I also want to congratulate you because for the presentation, I mean for the how it's written and the format, and I think it's very clear, and especially the introductions are very well written, and your presentation has been also very clear. My only two comments is first a general comment that sometimes the conclusions are too short, too concise, and there are more. Normally it's the other way around. I think the conclusions people try to exaggerate a little bit the result, and you are doing the opposite. That sometimes you read the conclusions and you think there is not so much, but you go to the chapters and the results are really relevant. So and another comment on the thesis is that that's a small comment, but I think it's important that in the introduction that I think it's very clear. When you mentioned you are always working with multiplicative noise, and then you mentioned the Ito and Sakonovich interpretation, and you say, well, the conclusions are not so dependent on the interpretation, and but it is. I think it's not true. Especially in collective effects, it's for multiplicative noise is very relevant in many systems. Of course, in the whole thesis, it deals with French noise, and then the interpretation there is no need of interpretation actually. That in other systems with white noise in time, I think it's very relevant. And now some questions on the defense. So first about the drug absorption, I would like to ask you a couple of things. The first one is that you call essentially the advantage of your method is that you don't need the nonlinearity. You code the whole nonlinearity in the equilibrium state and the frequency. Am I right? So you don't need the explicit expression of the flow, J.
It depends. The
omega,
the frequency and the friction will change according to the selected nonlinearity. So in fact the so here I mean the approximation was first this with the friction. So here into the friction it goes in the derivative of the nonlinearity.
Yeah, but I mean, if you want to apply your idea to real data, you can just consider the equilibrium X and the frequency as parameters.
The thing is, the thing is that the equilibrium concentration is not measured, never, in the experiments. Apparently, this would take too long, and I think it is maybe, I don't know, because it is too many errors, I don't know. They basically never measure. We actually asked them if this was possible, because I think it would be much better to derive all these parameters, but apparently they never do so. And I mean, this is the first 2 hours. So this is the linear regime of the slope of the first 2 hours. And I think, I mean, here I said that the equilibrium is reached after, like, this would be 60 hours. This is far too long for, what they do is they have, like, I don't know, hundreds, dozens of these little pots with different initial concentrations, with one side or the other side. Of all those, you have to do 6 experiments or a few to have a mean value, and this is why they only rely on what is it? On this measure, and the equilibrium concentration is not accessible. So,
and didn't you think of having noise or studying the effect of noise or the diversity in this type of?
We did. It was a bit. We did think of it. I did actually as well start some calculations, which in the end didn't. Well, they didn't. Ended up there. They are not done. They are not finished. But basically, what I said about this in the beginning about these cells, that it is considered to be all equal, and this is why you can make this approximation. If you now say they are not equal, then you would have to ask what is not equal. So say one thing is the geometry, the size could change. The other thing is that I was thinking the expression of this protein, of the transporter protein, it could be different from cell to cell. So you would have these two parameters from the nonlinearity as well different. And then you have quite a few parameters where you would actually have to add this perturbation or diversity. So I started doing this to write down these equations, how they look like, setting, well, this would stay the same, as you say they are still mixing, but then instead of having one cellular volume, you say you have a large number of cellular volumes, and then devise different parameters. But this didn't bring any results yet, not any concrete results, but it is in fact possible to do so. And then one would have to see, I mean, the idea was a bit to see, if this, maybe it doesn't even, well, to see if it makes a difference or not. Maybe it is already sufficient to consider this 3-compartment model. Another thing is that as the intestine is a long tube, so this will not be constant along the whole absorption process. So I think there's quite a lot of possibilities to extend this model and to introduce this, and even noise would be possible to introduce. We actually didn't do that, which is maybe a part which is maybe missing and would enclose into the whole thing a bit more.
And now, going to the next chapter. But at some point you mentioned that it's because of this, you had this explanation of the oscillator then. So